everyone. Welcome to All Things Indian Friendly Voices. After a few episodes on history, music, and a little bit on pandemic and vaccines, in this episode, we are going back to science. 20th century was such a boon for all kinds of development, growth, evolution of science and technology. Some of the aspects are very well known. But despite all these things, I mean, irrespective of what all the developments are happening, and there was always a kind of a feeling that all these developments have not allowed or have not enabled human beings to understand themselves better. And partly because even in 2021, the amount of knowledge that we possess of human brain, is pretty limited given the development, advancement, everything, tools at our disposal. When Watson and Crick developed, they wrote a paper more than 60 years ago and presenting us with the DNA model, double helix. And that, it's like it started a chain reaction. A lot of things happened. In addition to genetics, various branches of genetics took place. And we have 30 years ago, when John Franklin brought out, this is all, I'm a lay person. So today we have a very good friend who is a professional. And when John Franklin brought out that molecules of mind, and when he proposed all mental illness can be resolved through chemicals, molecules, and purely brain. So although ironically he titled his book as a mind. So basically it's all towards the dispelling the myth of mind. And then we have all of you, most of the followers of our channel on Twitter, we all have gone through, debated Matt Ridley's genome. And today we have come, all these developments apart, we have gone further ahead. And one of the things is the offshoot of all these development is that a field of study called neurogenetics. That's a topic today. It's basically a kind of a field or research, in part, I'm just putting in a very simplified generalist term. Basically, it's a kind of a look into or present genetic basis of normal and abnormal, what to say, abnormal nervous system, like nervous disorders or how the nervous system functions. So I'll ask Suresh to introduce our guest.
Yeah, thank you, Rajmohan. And first, let me welcome Dr. Surabi Veeraraghavan. Welcome, doctor, to the show. Surabhi Viragaman is an assistant professor in the Department of Molecular Human Genetics at Baylor College of Medicine. This is located at Houston in Texas, USA. She is also the co-director of rodent neurobehavioral core at the Neurological Research Institute. This again at Texas. It's at Texas Children's Hospital. She got the PhD from the same school that she's now the assistant professor at, that is from the Baylor College of Medicine in Houston. Before that, she had completed her master's from Ramchandra Medical College in Chennai and her bachelor's from the Stella Maris College in Chennai. Her current work is focusing on the development of what is called as a preclinical endpoints and developing novel therapeutic strategies for neurodevelopmental disorders, including several syndromic forms of autism spectrum disorder. Now, don't be worried about all these terms because Dr. Surabi is going to explain all of them to us soon. I must also mention that this is the profile I got from Dr. Surabi, but beyond that, I know that she is a very multifaceted personality. She is a trained Bharatanatyam artist, and she is a fabulous cook who has her own channel running in Instagram, and she's also a sort of an inspiration for a lot of people on Instagram, where she has a good number of followers and where she speaks about various issues, including mental health. That's a background. So we today have a very good personality with us. And of course, we are going to talk to her more about the technical part of her work. But as I said, she's much more than what her technical work is. So to start this whole conversation, I will ask Rajmohan to take this up and begin our discussion with Dr. Surabi.
Yeah. Thanks, Suresh. Dr. Surabi, basically, I want you to start start telling us what this field neurogenetics is, because I remember there was a guy on Twitter. I don't know whether he's still there. When we were all discussing, some doctors also jumped in. They said this, etc., and then CRISPR has come, blah, blah, blah. And that fellow asked a very simple question. That is a deadly question. People laughed at him and threw him out, especially the doctors. And he asked a very simple question. Okay, if hypothalamus is what is controlling all your stomach, he gave example. Why don't we just put a button switch? Why don't we open our brain and control the whole body through brain? So then somebody tried to explain, and at that time, 5-6 years ago, we got to know about there's a term called neurogenetics. So please tell all of us, enlighten all of us what this neurogenetics is.
Sure. First of all, thank you so much for having me in this forum. I'm very excited because I usually get to talk about more broad generic mental health things, but this is very specific to what I'm working on. So this is great. And thank you for that very lovely introduction. And like you mentioned, so neurogenetics is a field where we are trying to understand the role of genetics in the functioning of nervous system. So when I say nervous system, it refers to both your central nervous system, including brain, and also your peripheral nervous system. So understanding how our neurons and neural circuitry develop and how it manifests in itself in terms of phenotypes. We use this word called phenotypes repeatedly in this field. And what is this phenotype? Phenotype is what you see when you observe a person. Let's say I'm looking at you now. You exhibit certain characteristic features. Now those become your phenotype, your behavior. So that is very important because it's very hard for us to open up the brain and see how the structure of the brain is altered. So what we see, either physicians or scientists, is what you manifest, right? So in the field of neurology and neurogenetics, this term called phenotype and behavior is very important. So when you specifically come to neurogenetics, what you're doing is you're trying to understand the roles that genes play in controlling behaviors, in controlling neural development. Now, if you look at the term neurology, a broader term of neurological diseases, they encompass all disorders of the nervous system. Now, it can be disorders, something as simple as your migraine, stroke, or anything more physiological. Or you can go to the other end where you have your neurodevelopmental disorders, neuropsychiatric disorders, neurodegenerative disorders, all of which have a very, very strong genetic component. So all of these come under this big umbrella term of neurological disorders, right? Now, we are trying to understand what are all the genes that play an important role in governing or in these in different neurological conditions I just mentioned. So when you say neurogenetics, it's a very, very broad umbrella term, trying to understand genes that govern these different illnesses. So that's how we look at it.
Okay. So essentially, when
one second, Suresh. Is that that means from what you were saying, I understand phenotype is an expression.
Yes, exactly. So phenotype is how your body manifests it.
Okay.
So if I have a cough, that's a phenotype because I'm manifesting whatever internally that's happening in my body. I'm manifesting it as a particular behavior.
Okay. I say, okay. So a twitching of the eye or anything else, suppose
exactly.
You are in.
Right. Because the problem is when it comes to neurological condition, unless it's something like stroke or spinal cord injury, where you can physically in a scan or an MRI see what's happening in your brain.
Okay.
For majority of the conditions, let's take schizophrenia, bipolar disorder. How do you actually see what is really happening in the brain or spinal cord, right? It's more internal. So how do you even understand that? You understand the disease by looking at how so for a lot of neurological conditions, studying genetics means you're trying to understand how each disease manifests itself in people.
Okay.
So that's, and to give you a better example, if it's a heart condition, you have very clear markers. Cholesterol, I mean, I'm just randomly giving you some numbers, looking at blood pressure, cholesterol, like you have very, very clear numbers that you can gauge, where you can say, okay, this is a high range, this is a low range, the person has a disease. But in a lot of brain-related disorders, you don't have such markers. So how do you understand them? By looking at the behavior.
Okay. Thank you. So essentially, that's why I think the term has come. It's all in the genes, right? When they say, right, if the person is very good at something, they'll say, it's in the genes or something else. That's something which I also wanted to ask you. Does this also mean that you study how this transmission of anything happens? I just have two parts of the question. First is as an extension of what we were speaking, in the sense that a lot of things we hear, again, very layman terms, like, these are transmitted through generations, right? Like for example, let's say, as you said, a very common thing like diabetes. So if mother has it, the chances of you getting is very high type of thing. So is there anything else that we look at transmission? Second is like, who are the pioneers in this field? That is, how did this field develop? Because initially, I think there were two different things, right? Doctors studied neurology, and the biologists probably studied the DNA and others. So how did they combine together?
Yeah. So let me address that first, and then we can go to your first part of how it's transferred, because it'll make more sense if we go that way.
Sure.
So I think pioneering work happened in 1950s and 60s. So around 1950s, the field of genetics had already started developing a little bit, because in 1950s, Watson, like they already came up with the whole idea of double helix DNA. So there were a lot of progress, and the concept of gene, the concept of inherited disorders, I mean, that had already started developing. So there was progress happening in the genetics field. And then there was also progress happening in the neurology field independently. People studying brain disorders. But in 1950s and 60s, there was one scientist who played a very critical role, Seymour Benzer. Now, you can call him, a lot of people call him like father of neurogenetics, but the idea is he was one of those earlier scientists. There were other people too, but he was one of those earlier people who understood that genes could be playing a role in behavior or brain-related functions. So he was actually a physicist who branched from physics to biology. It was a very interesting story. If you read his life history, it's quite interesting how he jumped from physics and those principles and applied those in biology. So anyway, he worked with bacteriophages. Then he did a lot of work with Drosophila. And that was the model he used to try and understand the role of genes in behavior. So it was one classic experiment he did where you have a vial full of these flies and you bang the vial, these flies start migrating towards light. So that was a simple experiment he did to understand, okay, this is interesting behavior going on. Let me see if I can like tweak this behavior using different Drosophila mutants. And he was able to do that. And so that's kind of how the whole field of neurogenetics kind of started.
Wow.
And then, of course, a lot of progress has been made in the last 30, 40, 50 years. But I think a hallmark event would be the whole genome sequence, the first draft that came out in 2001. That kind of, I would say, revolutionized the field. So until that point, right, people were like thinking, neurologists were working independently. You had like geneticists working independently. But now that there was so much advancement happening in genetics and genomics, clinicians and scientists started slowly coming together in trying to provide more holistic, personalized medicine. So I would say in terms of like personalized medicine and how all these fields merge in the last 20 years, a lot of work has happened.
Okay.
But I think that really, I think 2001 again played a very critical role with the genome sequence. We kind of understood how many genes are there, and it was easier for us to look at diseases from the lens or through the lens of genes. So that's kind of how this field has evolved. And I think today where we are is we are at a stage where personalized medicine is going to be the future, where you are going to have to, if you're a clinician, you have to understand genes because they have now become inseparable from disease mechanism. I think we've all accepted. Earlier on, it was hard for clinicians to accept or understand the idea of genetics itself, right? When I say early on, I'm talking about 15 years ago when I was starting my bachelor's and master's. When I was doing my master's program at Ramachandra, we had a human genetics program. This was in 2003. Even at that point, clinicians were finding it very hard to accept that, okay, genetics is the field that I actually have to like pay attention to because it governs disease management. It was still a little alien, right? A concept because human genome sequence had just then, come out. But I think in the last 20 years, we have made tremendous progress in terms of marrying both fields, and now they are inseparable. You have to look at neurological disorders, or in more particularly neurodevelopmental, neuropsychiatric, and neurodegenerative diseases. You have to look at them from the point of view of genetics, because otherwise it's going to be very hard to understand. So we have reached a stage where everyone's accepted that genetics is the future. That's how you have to look at medicine. So that's where we have evolved now. And we started off 50, 60, 70 years ago, kind of. It's been a nice 60, 70-year-old journey, I would say. Also, one other thing I want to point out is another aspect to this is advancement in computation. And I know because you're from a computational background, I think we have also been able to make tremendous progress in genetics and genomics only because of like the computational progress that the world has seen. Because in 2001, when they sequenced the genome, it took them billions of dollars. They had to do like really age-old shotgun cloning methods. It was a very, very long, cumbersome process. But today, 20 years later, you can get your genome sequenced in less than $1,000 at a very, very, very fast speed, right? And all of this is possible because of all the computational advancements we've had. So I feel it's not just genetics and biology. It's only been possible because of computational advancement. So that's also what I'd like to mention.
Yeah, like you mentioned, things are merging together. Like, those initially what you would think that, as a part, we will let this subject go as a part of a choice, or elective is no longer elective. It becomes like mathematics is now permeating everywhere, especially if they get into big data and others, right?
Yes.
So you cannot anymore say that I'll take a biology because I hate math, because you will end up with math sometime or the other.
Absolutely. I think the time has come for us to actually, I mean, I don't know if this is relevant talking in this context, but I just felt I should mention this, but I think that a time has come where we really have to look back at how we train our high school kids and undergrads and programs, because we no longer can do, group 1, group 2 and all those things. All sciences have merged. Like you rightly said, if you don't understand maths or don't understand computers anymore, you cannot do genetics. Yes. You cannot do, coding is the future, and you have to understand coding to actually understand genetics. And because like you also mentioned, big data, we have all these tons of sequence data. How are we going to make meaning out of it, right? So I think now genetics, biology, mathematics, computers, everything has merged. So I think you gotta like kind of look at it as a complete.
Dr. Surabi, you mentioned something which is 2 words. One is genome sequencing. Another is personalized medicine. Now, what are the advantages? Can you please tell for the benefit of everybody? what are the advantages of somebody who is spending $1,000 and getting this genome sequence? What are the advantages for the individual? Is it like something like that some time ago, some 23 and me came, all your history? I mean, is it something like that? Somebody who's offering $400, people may go to quacks. Okay, so that is one. Second is that personalized medicine. I mean, uh, especially some of the, when it comes to mental health, main order or disorder, personalized medicine will it not be dangerous? Ethical issues are there. People can manipulate. I'm not asking something out of some murder mystery from Netflix. I'm just, when you are talking, it struck in my mind. So two questions. What is the advantages for individual for genome sequencing? And second is ethical issues concerning personalized medicine.
Okay, one quick interruption here is first, can you explain what is meant by genome sequencing and then what is the
Yeah,
advantage
okay.
of it? So,
Okay.
Sure.
so you can
So
start
let's
from
go in that order. So when I say genome sequence, what I refer to is basically getting your whole DNA sequence, your chromosome, your DNA sequence read. So when we say DNA, there are base pairs, right? So ATCG. So your genome sequence, what we do is when you, like, if you think of a cell with DNA in it, you can't like have one machine just reading your entire sequence in like one stretch. You have to cut it into smaller pieces, and we have sequence reads, and you read each sequence. And now we have great computational tools that can very easily put together all the sequence and give you like a full sequence of your genome.
Okay.
Now that's how we do sequencing these days, and that's how it's very fast because of computational and bioinformatics development. Now, to come to your question about what is it that you gain out of sequencing your genome, right? Now, like you mentioned, sure, you can use, there are several services, right? 23andMe, several services where you can just give a sample, get your sequence read. Now, you're going to be given thousands and thousands and thousands of base pair information. What do you do out of it? And before I further explain this, we must also understand that all of our humans, I mean, all human beings are very unique and different. Yes, we share genetic information, but we also have unique base pairs called nucleotide polymorphisms. So just to give you a very, very layperson example, maybe in one particular position in my DNA, I might have a G, but you might have a C, but both of us might be normal. Having that polymorphism does not always mean you have a disease, right? So what they did was when they did human genome sequencing, they have a control sequence that they compare it with. And then they compare it with that and they say, okay, you have these mutations in this gene, so you might be predisposed to these conditions, right? Now, this is crazy. It's a crazy approach if you don't have a specific question in mind. Let's say you get your data. What are you going to do with the data? Okay, C, T, G, A, T, G. What does it really mean? And I'm pretty sure there are going to be hundreds of polymorphisms, all these things in your DNA. Does that mean you're going to get all 100 illness? No, because there is this genetic information. Plus, you have something called an environmental insult. You have to have a genetic insult, environmental insult, interaction, manifesting into a disease, especially with neuropsychiatric conditions, mental health disorders. It's not as simple as, hey, there's a mutation in this gene. No, and then the person gets a disease. No, there's a lot more. There are a lot more steps involved, and it's a lot more complicated. So I wouldn't recommend this for anybody. Like, if I've never gotten my DNA sequence, and I will never get it done, even though I'm somebody who's in this field, because I don't have a need, a question to get it done. I think to get your genome sequence, you have to have a question. Let's say you have a family history of breast cancer. And breast cancer, BRCA, we know for sure that it is there's a very high chance of the child developing it if the mother has it, right? So in those cases, sequencing makes sense because you're specifically looking for a mutation in a particular gene. Then makes sense. You sequence it, you look at that particular genome. Okay, there's a mutation. You're already put on, maybe you start your mammograms earlier on, right? So there's a clear answer to it. unless you have a question, it's like needle in a haystack kind of thing, right? Like you will be lost completely if you don't have a clear region you want to look at. Because you're talking about 35,000 genes, like you will find many, many hits. It will not make sense if to just generally look at it. So you have to have and it's also not something, at least in the US, your insurance doesn't cover you a lot of times. And you have to have like a diagnosis for you to even go and, do it, unless you go to like a random 23andMe kind of website and just pay money and have it done, right? So to answer your first part of the question, you have to have a question of what you're looking for in your genome. Otherwise, you will get so much information which won't make sense to you. And the second part is the ethical question. And I completely agree with you. The problem is randomly getting your DNA sequenced is because you will have a lot of information that are more detrimental than useful. Because I always like look at it, I'm also a little philosophical in this, and this is my opinion. This is not what the field believes in, or it might not be what everybody believes in. For me, unless I have specific things that I can do to prevent a disease, I don't even want to know I have a mutation. Meaning, let's say I have a family history of certain illness, and I find out I have a mutation that puts me at a higher risk of developing, great. Then I can modify my diet, I can modify my lifestyle and prolong the onset of the disease, right? Push the progression or the onset of the disease. Then it makes sense. Otherwise, randomly sitting on this piece of information that tells me I have a mutation in this gene, mutation in this gene, mutation in this, which means I might get some 10,000 different diseases 5 years from now.
So very difficult.
But
almost like, I was just saying, it's almost like people having some symptoms and trying to look and Google to find out what it is, right? They'll end up thinking they have 100 diseases at the same time. Right.
Yeah, exactly. That is from the subject's point of view. But from the, even when it is such an information, let's say when more and more, as the days go by, as Dr. Surabhi said, Suresh, that this personalized medicine, more or less, we are going to have some chip which will have all our history very soon. Correct. I'm not saying that it will be in our body, maybe in a mobile. Somewhere it will be there, and all we have to flash. Maybe like a credit card, we have to just flash it, and then the hospital will give a date and time, and the doctor need not ask us questions unless he wants to ask specific questions like what symptoms, what brought you here. So that when you have such a vast amount of information in the hand of or like medical professionals also, it could be proved dangerous.
Yes. I agree with you 100%, because the problem there is, I'll give you a simple example, is in US, insurance company, when they know you have a predisposition for a particular, condition, your insurance rates really, really go up, or you might not even get insurance, right? So a simple example. So if you have the genetic information in your chip, and they use your chip to get all the information, they find mutations in genes that puts you at a high risk of diabetes, they're not going to either give you insurance or your rates are going to be very high. It's very, very unethical. Or people freely having this information. So I am actually, I am not pro that approach. For me, I think personalized medicine should still be confined to, let's say, a physician has this, he or she has noted this history in a patient. There's a need for sequencing. There's a need for understanding these genes to figure out what's going on with the patient. Then it makes sense. I think if we make it, I don't know, I just feel if we make it open like this with, let's say in a chip or something that everybody carries, we have to have very, very stringent mechanisms to keep things under control, because otherwise it's going to open a can of worms that I don't think we have the resources to handle. I mean, take simple things as social media, for example. We're discussing so much about WhatsApp and Facebook and privacy, right? This is like several layers beyond that, much beyond that. So I'm still a little conservative when it comes to personalized medicine, where I think it's great. I think that's the future, but I think it should still be used with a lot of restrictions. It should still be used with caution. I don't think it should become one of those.
And with boundaries for the information. I mean, where the information could could spread.
Absolutely. I agree with you on
Yeah,
that.
my next question actually is about same DNA. But before that, I just wanted to quickly ask you. You said in the sequencing you're talking about ATG types, right? Are they the amino acids or something, or is it
They're
something else?
nucleotide base pairs.
Okay, okay.
Yeah,
So no, I just sequences just how they are in your body. So
correct.
essentially, from what I understand, again, most from a layman term, from what you said about gene sequencing, is that everybody seems to have a different uh, combination of these ATG or whatever you were saying, right? These amino acids or whatever the basic material, correct? And that this combination is tested against some baseline. They have
Mhm.
decided this is a baseline, and then you see at which place the in the sequence you are. For example, they have something called A, and you have something called T. Is that something? Okay.
Correct.
So, but
Right. For the
but
most
because
part, you will share, for the most part, we'll all have identical
identical
sequences.
stuff.
It's
Correct.
only when in these sites where there is different polymorphisms. But the
Correct.
idea I was trying to say was not all polymorphisms lead
Results
to
are bad. Yeah, okay. Not all polymers bad need to be bad. Okay. Okay. Now the same, because we're talking about DNA. When I tried reading up something about neurogenetics before coming to talk to you, I kept reading about this term called recombinant DNA. Right. It seems to be the major thing and one of the ways in which genetics works and all that. So, what is this recombinant DNA, and how is it used in your field?
Right. So, in general, recombinant DNA, I would say, is an idea of you manipulating the DNA, the genome, the way you want,
Okay.
and you're putting it back into the system. So that's like a very, very broad way of defining recombinant DNA. So you're making the manipulation that you need in the DNA, and then so that's when the whole word of cloning comes into picture.
Oh, okay. Okay.
So this technique is very, very popular in medicine for generating proteins that you cannot generate in your human body. Let's take insulin, for example. It's a great example. You are taking cells, it could be any kind of cell lines, bacterial cells, whatever medium you take. You take a plasmid. Plasmid is a circular DNA. You introduce whatever gene, you introduce insulin gene in there. So it starts producing insulin.
Oh,
And that insulin is extracted and injected in patients.
oh,
So
okay.
recombinant DNA is a technology that you can use to generate certain proteins
Mhm.
that in turn you can use in
Okay.
humans.
Okay.
So broadly in medicine, it's used a lot to generate a lot of different molecules that your body needs, that you produce them outside and then you purify them and then you can inject them
Insert. Oh,
your
okay.
body.
Okay.
Yeah, yeah.
Yeah,
So that's
Rajmohan.
Yeah.
yeah.
So, I mean, continuing with what you said and what Suresh asked, and well, there is something like, initially you said that neurogenetics is about disorders. Your study of, neuro disorder, the role of genes, genetics in that. I mean,
Mhm.
is there a positive side to it? Like, for example, 7 years ago or 8 years ago, a television series in America came. We were all just laughing, actually. I mean, it's just far-fetched, gene editing. So that the doctor in that, I don't remember it came, it was streamed in Netflix also, that that doctor says that you edit the genes to prevent a disease, somebody from getting the disease. So what one person in the lab, he will try to do a lot of mutations and try to figure out and come to some kind of a finding, discovery that will make human beings disease free, whichever virus comes, so like that. But in next few years, we all read in newspaper and TV on the internet, oh, CRISPR is there. And so is there a positive side which is completely, totally into this prevention of diseases?
Yes. So yes, but it's not as simple as what they show in media.
Yeah,
So
Netflix, yeah.
yeah. The first aspect, let's talk about genome editing, and then I'll talk about therapy, gene therapy, and how
Yeah,
that
yeah.
editing phenomenon can be used in patients.
Yeah,
And
just one small entry. This
yeah,
thing is that you can also tell what are the major neurological disorders that you cover, and how this is used in that. Okay. Yeah.
sure, sure. Yeah.
Please,
So
yeah.
let's start with the whole basics of editing and gene therapy, and then
Yeah.
I'll go
Sure.
into like disorder. Yeah.
Sure. Okay.
So, so genome editing is like the in thing. Like everybody is editing all kinds of genomes. So so what you essentially do is you take a human gene, and again, all of these can be done in, let's say, cells or bacteria in lower organisms. You take your human gene, and then literally like imagine like it's all happening in like test tubes and like outside of your body. So you take your human gene, you can do base pair edits. So let's say in your mutated human gene, there's this one mutation in one particular, when I say mutation, changing of a base pair in a one particular region, you alter that, then you can take that and you can put it back into an organism. So that's the whole idea of genome editing. And we did a lot of genome editing when we were generating mouse models and rat models. So that's why the whole concept of CRISPR and making gene knockouts, overexpression models. So what we're doing essentially there is you make all these edits to the gene, and then you put it back in a mouse, and you create a mouse or a rat or a Drosophila, like mostly rats and mice, with that mutation. So you make a model of a particular disease in a mouse or a rat. And that's how we study, and that's how I do all my research. I have several models of rodents that mimic certain human conditions. And so that's how we study that. So right now, a lot of work is happening at the rodent level to edit these genes, try to see if we can put the edited gene back in humans, will it work, and all of that research is happening. And I'm giving you a very, very like a 10,000 feet bird's eye view of the whole thing. I mean, it's obviously a lot more complicated than that. So from genome editing, the next part is, okay, let's say I edit the genome in a mouse in a certain way. Can I apply that in the clinic? Because if it's not going to be useful, and if I cannot put it back in the clinic in patients, what's the point of research? And that's how in current days, gone are the days when we can actually do science for the sake of science. Now we have to have a significant section for every grant where you have to talk about how it's going to be relevant in a clinical setting. Otherwise, funders don't want to give you money to just, they want to know if there's going to be clinical relevance. So the next stage will be genome editing in humans. So that's why the whole concept of gene therapy comes in, where what people are trying to do is, can you inject a fixed gene into the patient? And our DNA, our cells, they are magical. You introduce a genomic piece, it will recombine with your own chromosome through various mechanisms, and it will actually start producing normal protein in your body. I mean, our cells are just magical. I mean, just from like a scientist perspective, I find this whole thing so really magical, right? So here you're trying to inject the piece of DNA into humans and see if that fixes it. Now, great example would be cystic fibrosis, where there's been very, very successful gene therapy trials in that field. Now, coming to the field of brain, has gene therapy actually worked in patients, which is this editing phenomenon worked in patients? Slowly, we are starting to see some success. Now, the first example would be in Angelman Syndrome, where what they're trying to do is, so Angelman is an imprinting disorder where you typically have two copies of chromosomes in your body. One is silenced. So the protein is produced only from the other copy of the gene. Now, in patients with Angelman syndrome, the copy that's supposed to produce the protein is also mutated. So one is silenced, one that's supposed to produce is mutated. So the patient literally has no protein for that particular gene. So now with gene therapy, what they're trying to do is, can you introduce a DNA molecule that will combine with the antisense molecule that is silencing the gene, release the silencing, so that the silenced DNA can now start producing protein, right? And they have actually been successfully able to do this in the first and second phase of clinical trials in Angelman patients. Of course, there are lots of side effects and the data is still coming out, but I can totally see how in the future this might be helpful. But again, every gene therapy has a caveat, and with brain disorders, there's a larger caveat, which is we have no idea if it's going to work in every single patient. We have no idea if this is going to work for all neurological disorders. We have literally cracked, like, this is like a baby first step that we have taken into the field. Like, there's a lot more that we don't understand, we don't know. But to answer the question in a very simple term, yes, it's possible, but we still have a long way to go in terms of actually seeing this in widespread in a lot of patient population. It's going to take a lot of time, but it's possible.
So before I ask my question, a small clarification here. See, when you say you take something out, you do some recombination or gene editing, and you put back that gene, right? So in our body, there must be a lot of genes, right? A lot of the genetic material, there's huge amount of it. So when you put this one, right, how does this impact? Because they're already, the other genes are already there. So how does this new gene impact you? Because is it just like, in a million, you're just introducing 1 or 10? I'm just, I mean, I don't know if I'm thinking right, but.
It's a great question. And that's why it's important that you figure out the site of injection. So when I do, do a lot of these gene therapy studies in my lab, where we have to specifically choose which part of brain we inject so typically these are viruses that carry, so you need something that carries the DNA. So we choose viruses that carry the DNA we want to introduce into the body. So we have to be very careful of, am I going to inject it in the hippocampus, in forebrain, like where am I injecting? So you want to be very specific about where you inject, because there's only certain region that your virus can spread. And also, if you're looking at brain disorders, a lot of these proteins that eventually get produced, maybe they won't cross the blood-brain barrier. Like, we don't know what happens, right? So it's very important that you inject it in a place where you can have maximum effect. If it's a brain condition, the hope is, again, this is us assuming that if we, let's say, inject it in the hippocampus, for example, maybe that is sufficient. Maybe that's sufficient to fix some of the symptoms. And the reason I'm saying it's highly complex is because it's not as simple as, okay, I'm giving this injection into this patient, it's going to fix all the symptoms. No, because hippocampus is maybe going to control some symptoms in the patient. There are so many other things that you cannot control, right? So we are trying to maximize the effect, but we have no idea. The answer is we don't know. But we want to pick a region that will work the best. And it's you have to try. I mean, that's why we use a lot of model organisms to test and try everything. And there's no guarantee that that's going to work in patients. So there's a lots of ifs, and if it works and if it does not, a lot of that. But the concept works. The concept of fixing and editing DNA. In fact, people are even thinking of editing your genes when the baby is in the mother's womb, right? Wow. So the concept works. We have proof of concept. We have that. But it's going to be a lot more complicated actually implementing it for many diseases, because brain disorders are way too complex. So to actually kind of tie this all together, I think Rajmohan, I think you mentioned at the very beginning that we have made a lot of progress in terms of
very little,
that is very true. Even though we have made a lot of progress genomics, genetics wise, and a lot of progress in neurology wise, we still have a long way to go just because of the nature of brain disorders. Now, if you look at cancer or cardiac diseases, we have made a lot more progress just because there are very, very defined, clear biomarkers, endpoints, etc. So brain, it's way too complicated. So we have to catch up a lot. Even though the progress has been made in terms of discoveries, we still have a long way to go just because of the nature of brain and how it controls everything and how complex it is. So that's
Dr. Surabi, just this is not, it's not a question, just an observation, because the question Suresh asked and the answer you gave. About 15, 20 days ago, I met with an old man. Okay, when I was young, his father was very old, 86 or 87. So whenever he gets some kind of a hallucination, that suddenly, used to be very different. The manifestation each time was different. Sometimes he will become scattertronic completely. Sometimes he will just come around the chair. So sometimes he will just remove the clothes and stand in the road, roam around. So all that this man, when I went and talked to him about the development of all these things, exactly this point which Ed Yong or Paul Zimmer, somebody wrote in New York Times, I think, that we have understood only 10% of the brain. 90% is a dark matter. So when I said, what, don't believe all these people. All scientists are all because, and he said, 25 paisa largactyl will do. His impression is that. He's not a trained this thing. Yeah, that's true. Chlorpromazine is a wonder drug. Although it was for hallucination, it also for a very hyper patients. It used to just, you to spend 25 paisa, a person becomes normal for a week. So he said, what is all this? They are spending crores of rupees on all this. He was telling, I just recollected that. That's all, yeah. So please go ahead.
It's a great example to describe the field, actually, honestly speaking, right? The thing is, what is it that we are trying to achieve? Now, for brain disorders, we already know it's highly complex. Let's take schizophrenia. We are talking about so many different behavioral problems in this patient, right? Now, we are trying to identify, so right now what is available for something like schizophrenia, or let's take autism spectrum disorder, which is what I'm working on, which I'm more familiar with the recent developments, right? In autism spectrum disorder, every child manifests itself with many different behavioral problems, social problem, communication problem, many things, right? In terms of therapy, right now, what is available for these children? Either behavioral therapy, or we take an old drug that has worked for other diseases, and we give the drug to the child to take care of specific symptoms. Let's say the child has seizures. Then we give a random seizure drug that has been used for other seizures and give it to the child to control specific symptoms.
Mm-hmm.
But in the field, what we're trying to achieve is we want to understand the mechanism well enough so that we can maybe come up with one drug or one treatment option that will literally take care of multiple domains.
Mm-hmm.
Because right now, I have met children who are on so many meds because you take one medication for seizures, you take one for hyperactivity, but then those meds interact with each other. The child is a complete mess at the end of the day because you're talking about side effects of each medication that you're taking, right?
Yeah.
So the field wants to understand the disorder well enough so that you treat the source, and that hopefully will take care of multiple symptoms. So you treat the gene itself in terms of gene therapy, or you treat a one master regulator that maybe controls different pathway, maybe that will control multiple symptoms. So that is what the field is hoping to achieve. But are we there? No. We have a long way to go just because of the nature of these illnesses. It's way too complex, and not two child shows the same phenotype. For some children, because it's a spectrum, right? Some children can have mild, so it's very complicated, but we are making progress. It might not be at the same pace that we would like to do, but I'm hopeful that maybe not in my lifetime, maybe in our lifetime, maybe the next generation maybe might benefit, right? So that's the hope when we do science, right? Maybe today we might not see the results, but I am very hopeful that the future looks a lot brighter.
Okay. Just to put that also, as you said, you encapsulated the current state of the field very well. That was one of the coming questions. So I'll skip that because you already wonderfully explained the whole field and where you are. Just becoming more personal in the sense that in your own research, right? What sort of research you do, and what sort of success have you achieved, and what do you see the field developing?
Yeah. So I work on neurodevelopmental disorders. Now, what are these? These are early onset neurological conditions where it starts happening when the child is developing in the mother's womb, so that you see these behaviors as soon as the child is born. For example, autism spectrum disorder is a great example where when the child is born or when the child is very young, you'll start noticing that the child is not hitting those milestones. The child is not developing like any other
Speaking,
children.
yeah, okay.
Speaking, communication, eye contact, many
Correct.
things. Right?
Mhm.
So I study these. That's what the big term is, neurodevelopmental disorders. Now, under neurodevelopmental disorders, there are many different examples, right? So I focus on autism spectrum disorder. And under autism, you have syndromic disorders, non-syndromic. So autism is again an umbrella term that is used to describe children with social communication problems, repetitive behavior, and so many other aspects. Now, syndromic autisms are disorders where there's a syndrome that's already defined, and these people share symptoms of ASD or autism. For example, Fragile X syndrome, Rett syndrome. Now, these are syndromes where you specifically know one gene is mutated that's causing this disorder, this syndrome. And these children who have these syndromes all have ASD or autism spectrum disorder.
Okay.
So autism is more of a condition which can occur in other like syndromic cases, or you can just have autism without having a syndrome. So you have like two big categories, right? So during my PhD, I worked on Fragile X syndrome. Now I work on multiple things. I work on imprinting disorders, many rare genetic disorders, many different things. But big picture wise, what am I trying to achieve, or my group is trying to achieve, is that we want to identify two things. One is biomarkers in your body that can predict or that can tell you if you're going to develop a condition or not. Okay, biomarkers. The other thing is preclinical endpoints, which is what kind of endpoints should I be focusing on and studying that will be relevant to patients. For example, and I use rodent models, right? So for example, if I'm working with a mouse model of Fragile X syndrome or mouse model that shows features of Fragile X syndrome. Now, if I'm studying seizure and if the patients don't see seizures, what's the point, right? So you want to develop preclinical outcome measures that are relevant to patients, because then let's say I test a drug that works in my mouse model or rat model. When I take it to the clinic and give it to patients, it'll be more relevant in a patient context, right?
Yeah.
Also identifying biomarkers, because let's say in a mouse model of a particular disease, there are certain proteins that are elevated. Maybe I can just draw a patient's blood and maybe see if those proteins are elevated. If they are, then you can start giving the patient certain kinds of treatments, right? So looking at biomarkers and looking at preclinical endpoints, I think are both things that we are interested in doing. And our group, I mean, our vision has changed drastically over the years just because of how the field has evolved. Now it's no longer independent person working on independent project. It is all team science. It is all team science. So we work with clinicians who tell us, okay, this is what I observe in this patient for this particular disease. How do we address it in the lab? So then it gives me a reference point saying, okay, I have to only study seizure because that's what they really care about, right? So you need that feedback. Otherwise, all these years, we were studying certain things in models thinking they'll be more relevant to patients, but they're honestly not. And this happened to me a couple of years ago when I went for a conference, when I talked to these parents who have children with several of these disorders. And they were telling me that they don't really care about what I am working on, because what is more important to them was, let's say, seizure. But I was studying social issues, and they were like, look, if my child doesn't talk to another child, I don't really care. But my child has seizures every single day. He or she might die of the seizure. So I want that fixed. So that I was like, wait a minute. We are working in like two separate tangents that doesn't. So, we change our strategy immediately. And so now it's more of, we work with clinicians who give us data. We do the mouse work, the rodent work, the research. We also talk to psychiatrists, psychologists, patient advocacy groups. Now, patient advocacy group is a new thing that is integrating a lot into the mainstream sector in the US, where you have representatives who advocate for patients and tell us, look, this is what our patients want. Are you making progress in this? And patients give you valuable information. They tell you how their child is doing and all of that information, which you then can tweak your research. So your research becomes more meaningful when you get all these
integrated.
feedback. integrate. So it's all about team science these days. And it's not just coordinating with clinician research, it's also scientists talking to other scientists, which would never happen 50 years ago because it was competitive, right? Where I have to get my paper, I will scoop their idea. Now it's slowly changing, where you have working groups where multiple scientists come together and say, hey, this experiment failed. It's not working for me. Don't even waste your time. Instead, you do this, and then let's pool all the information. Let's give it to patients. So I think it's finally, I think people are letting go of their egos. And finally, I think I'm starting to see some change in trends where, people,
things are becoming better.
To be for different diseases that I'm working on, we have different working groups. We have different ones for,
Now, in your own research, I had this question because you were talking about this autism disorder. And this, as you said, you are working especially on those disorders which can occur right at the womb level, right? In the cases. So in these cases, suppose you want to prevent it, is there something which can be done to the mother's gene or something like that, which can prevent that such things happening? Or is it that after the child is born, then you take up the medication to try and control this disorder? Because I can understand a disorder coming up much later, you don't have a chance of controlling it. But in this case, is there a study done on why that disorder happens in the wound and things like that?
Dr. Surabi, just adding to that again, I go back to television or movie. Some time ago, there was a movie where a detective comes and he meets a biologist. The biologist says, oh yes, I mean, how do we get materials from? Because not only mother passes the genetic material to the children, but children also leave some of the genetic base in mother's body. So that from that we take, and then that has helped us to, I think it is kind of something related to autism. Because Suresh told me, I just suddenly remembered that scene. Yeah.
I mean, that part is true in the sense you can detect fetal material in mother's body. So sometimes they do that to test the state of the fetus. So that's possible. But in the context of autism, it's not that completely relevant. But for they call it prenatal while the mother is pregnant, if they suspect some kind of the child might have some abnormality, they have to take sample from the fetus, right, to test the DNA and chromosomes. Sometimes they can just take mother's blood because there is fetal DNA floating around in the mother, and it's possible to detect it sometimes. But in terms of autism, right, I mean, going back to your question about ASD in that context, autism spectrum disorder is very, very complex. So the short answer is no, but the long answer is it's very complex and we don't know. So there are many genes, over 100 genes, they say, involved. Plus you have layers of environmental insults. And they have to come together. And that combination is what results in these phenotypes. Now, many studies using mouse models have shown that when the mother is pregnant, when there's a lot of stress, for example, that can lead to autism-like features in children. And they've done many studies with mouse models where they stress a pregnant animal. The stresses in animal model could be single housing, confinement. It could be many different things that we use in like a lab setting. They have shown that stress could play a role in the pups born. You see ASD-like phenotypes. Because it's not ASD in animals, right? We always say autism-like or mimicking autism or something like that. It's different from, obviously, mouse thinking is different from how humans think. So yes. So we do know that stress when the mother is pregnant can contribute. But is it always true that and how do we read stress? How do we measure stress? How do we, stress is such a lovely term we like to throw around everywhere. Right? Okay. I'm stressed out, so I'm getting autoimmune disease. I'm stressed out. So it's one of those things that you can't quantify. But yes, the mother is not just contributing genetic material, she's also providing environment for the fetus to develop. So those 8-9 months of development is very, very crucial. And we are starting to understand that period is very critical. And to answer your question of do we know, can we do anything that can fix things during that time? For ASD, no. Right now, we don't know. That's because we have not understood what can be done. The thing is, yes, we can do things during development. That's the best time to attack, meaning you fix it at that time, then it'll get resolved. But which gene are we going to fix? What are we trying to fix? Because we don't have that information yet. So we don't know. We do know it should be fixed when the baby is developing in the womb. But what are we fixing? How are we going to fix it? What will fix ASD? We don't know, because there are too many genes, too many environmental factors.
Yeah, because I think another thing is, just as you are saying, I was thinking, even if you fix it at, say, the 5th month something, you don't know how it's going to develop till the 9th month.
We don't know. This is a complete black box. Only thing we know is intervention has to be early on. Early intervention is the key thing that drives all our research, because we want to catch it early, fix it early.
Correct.
But because in pups, right, we do some of these injections at P0, meaning as soon as the pup is born, we can inject. But how do you do that in a baby, right? There are practical considerations, and mouse timeline, human timelines are different. So it's a big black box, but we are slowly starting to
understand.
Starting to understand.
Okay.
Yeah, I think.
So, I mean, what is a going that you are saying that what is the success rate in terms of a transfer from experiments on mice to when it comes to human beings? Earlier, it used to be low. I don't know what's the success rate, especially for cognitive and neurological disorders.
I would say it hasn't dramatically increased. And I'm talking about specific neurodevelopmental, neuropsychiatric illnesses. Yes, we have medications that treat symptoms. So in that sense, we have made progress. But if you're looking at a medication or a therapy that targets the source and trying to fix it, we have made some progress, but not dramatic. Like, I don't have like dramatic success stories to tell you. Yes, we have made like progress in like Angelman's. We've made progress, in Rett syndrome. So we've made progress, but it's not gotten to a stage where it's completely available for the entire world to use. Like, we have not gotten there yet. But we have definitely made progress, but we still have a lot more to do.
Okay. Yeah. So one question.
Yeah. You had a question on the current mRNA vaccine. I think you had a question.
Yeah, 1 second. We'll come to that. Before that, I will ask, because it will benefit a lot of people. People are having like ADHD, which many parents are also very worried. So is there any kind of a path breaking, any kind of discovery progress there?
Yes and no. In the sense, yes, because we have understood how ADHD, manifests in people a little better. But again, in terms of treatment, we are back to the same position where we still treat children with ADHD using medication that are very generic medication, but we have not made progress in terms of identifying that magic pill or something, right? So yeah, so yes and no. Yes, we've made progress, but I don't think that has changed. Yes, it has changed the way we look at ADHD, because people are getting diagnosed with it a lot better. Our diagnostic tools have gotten better. Clinicians are more aware of how to treat children with ADHD, but we still have
some way to go.
Yeah. But I think good thing that's happened is in India, I feel there's definitely a lot more awareness. about autism, about ADHD, about seizures. Of course, there's always been work, but mostly ADHD and autism, I feel there's a lot of awareness, and people are going to clinicians, psychologists early on. So mostly for these conditions, at least in the US, behavior therapy is what's recommended. And I am, based on talking to like people in India, I know that in India now behavior therapy is becoming very common now, at least in cities, in big cities. So I think this is great because it's opened up a lot of avenues for parents who didn't have access to these things early on. So I think the biggest progress I would say is awareness in terms of diagnosis and treatment. So I think that way we have made progress for sure.
Okay, excellent. So the last segment, what we will do is now we've got excellent details from you about your work and the area and all that. A couple of more generic ones. First, of course, since you are in this area, and I know that you have taken the vaccine shot of coronavirus, which everybody is talking about. One is, of course, I want you to tell us what sort of side effects you saw just for our patients. We don't know when we'll get it, but given it is India, it could be faster than the US. First. Second is, they talk about this revolutionary concept of mRNA technology, right? What is it? What is it that is mRNA, and why is it different from the standard, the virus things?
Yeah. So yes, I've taken both my shots. I took the Pfizer vaccine, which is both Pfizer and Moderna are mRNA vaccines. In terms of side effects, I after the first shot, maybe a little bit of muscle pain at the site of injection. That is completely expected for any injection. After second dose, yeah, I had slightly more muscle pain and maybe slight elevation in temperature for a few hours. That's about it. Nothing more than that.
Okay.
It was completely fine. So that I think answers the first part of your question. And the second part, which is what is this mRNA vaccine? Now, traditionally, when we say vaccines, we take the virus, attenuate it, inactivate the virus, or take part of the virus and then put it in our body. So our body produces antibodies against, we call it the antigen, the viral protein. Our body starts producing antibody. Now, this is the first time where scientists, mRNA technology is not new, but using the technology for a virus and injecting it into humans, this is the first time. So mRNA technology has been around for the last 20, 25 years. People have been doing a lot of research. Now it's come to a stage where what we have done is, again, as a scientist, I'm amazed at how magical our bodies are, right? So here you are making an mRNA that codes for a viral protein. It's not the virus itself. You're just artificially making an mRNA that can code one of the spike proteins in the virus. You take that mRNA and then you put it in your body. You are tricking your body to make the virus protein. Then your body recognizes the virus protein as foreign and produces antibodies to it. So you're using your own body to make the virus, essentially, the viral protein. And then the body fights against it, which is, I think, like mind-blowing because of how amazing our body is, right? Where you just introduce an mRNA, and that's why also I think this vaccine is safe, because you're not really introducing an inactivated virus into your body. You're only inserting an mRNA that makes the viral protein, and your body generates antibody against it.
So what exactly is the mRNA?
So you have DNA. DNA we know is a genetic material. Now, DNA has to become protein because protein is the end point. Protein is what contributes to all the functions in your body. Okay.
Right.
So from DNA to protein, there's an intermediate step. And that step is your messenger RNA. Your RNA is what makes your protein. So your DNA has to become this messenger RNA that carries your genetic material. And that is what translates into a protein.
So you are making a specific messenger RNA which is capable of producing the virus.
A viral protein, a specific
virus protein. So essentially you inject only this messenger into the body.
Exactly.
And the body and the messenger inside your body develops the virus and immediately gets attacked by the body.
Yeah,
antigen.
so the body, it uses your own protein machinery in your body to make this viral protein. So
Okay,
you're
okay.
making this protein in your cell and you're
Killing it yourself.
killing it yourself. Exactly. So,
So that
in Tamil we say, right, one stone, two rendu ore kal
yeah, yeah, yeah.
Yeah, I
Yeah,
was
Rajmohan.
also wondering, you're right, like, when I saw that lot of hype, I thought this mRNA technology is there in Pune, Hyderabad, everywhere they're using. I was wondering why there is a hype, why people are jumping up and down for such a small thing. So anyway, now that you explained, first time they are doing it in a...
Like you said, the technology is old. The technology has been around for many decades.
Correct.
Using that for a viral injection, this is the first time.
Yeah. So one
Yeah.
final question, that...
Yeah. I think you asked one, I asked one. I think I have one
okay,
question.
fine.
Yeah, you go
Yeah,
ahead.
basically that where do you see the institutions in India taking up neurogenetics? Yeah, it's not much. Very little is there. Yeah, something is happening, but neurogenetics as a field of separate field of study, establishing research institutes or advanced study centers, that much is not happening at a big level. So where do you see, I mean, what are the things that needs to be done? Because India is all said and done, it's a population of 1.4 billion. So we are going to have, when you're getting diabetes, we are going to have maximum number of diabetic patients. Cancer means maximum number of cancer patients. Because at this rate, very soon India will leap beyond China in terms of population and therefore diseases also. So therefore we need more and more research into all kinds of fields. So what are the things that need to be done in India?
So again, I'm going to speak from my understanding of India, which might not be, I'm not like completely like up to date on all the institution that has this program, because I left the country a long time ago. But I completely agree with you in the sense that India has the perfect population to understand any kind of illness, especially when it comes to mental disorders. I think we can do a lot of things in India, because we have the patient population to study. Now, in terms of resources, I think India has the money. I want to believe that the kind of progress we are making in every other field, there are investors, there is money. I think what is happening is, again, this is a very naive understanding. I could be wrong because I don't know the current nature of education system there. But based on my understanding, I feel we don't have enough institutions that have programs that teach you all these different aspects. We are still studying zoology, invertebrata, chordata. Yes, that's important. I'm not saying that's not important. But fields have advanced dramatically. I think we need institutions where you teach them, like we were discussing earlier, you got to teach them genetics, biology, some bioinformatics, some genomics, and application oriented, which was my biggest issue when I was in school in India, because I read textbooks. But what's the point? We need to change our educational modality to focus more on recent research. Now, applying that in our context, because when I was in graduate school in India, I took up a research project where I was reading textbook and I picked up this project, but it had no application. I think we have to, maybe it's happening already, I don't know. Please correct me if I'm wrong. But my understanding, talking to people in some of these institutions, the impression I get is that we are still doing things, studying subjects that need change. I think we need to have programs that integrate all these various aspects and also bring in funding, so students who are doing these projects, PhDs, whatever, will have the motivation to work on something that has an application that can be applied here, right? So I think changes should be made at like the educational institutional level. And also, one thing I've noticed in the past, again, maybe it's changed now, is that doctors and scientists don't communicate. There's this huge, actually, even recently, I was trying to move back to India to try to see if I could establish something in the space of mental health genetic counseling, there is this huge barrier between clinicians and scientists, and they don't want to accept non-clinical professionals in the space of mental health. Like, there was a lot of pushback that people get in general for genetic counseling and stuff. I think those barriers need to be broken. We have to work as a team. So I see a lot of potential. I think for people in the future, like, if you, let's say, start an integrated program where you have clinicians, people like coming together, I think it's going to like have great impact in India. I think we have the talent, we have the money. Because I won't agree with the argument that there's no money in India. No, there are so many investors. People have so much money, and if you have a great proposal, people are willing to back you up, right? So I think I see a lot of potential. We just have to I don't know, like there's always red tape, bureaucracy everywhere, but I think we just have to figure out a way to like get past that. So I think it should start at the institutional level is what I feel.
Yeah, I mean, you are saying that what you are saying about this pushback attitude, and earlier you said these advocacy groups. I read somewhere that they went to a very, big hospital in Los Angeles, and then a doctor was there, and he just called, who are these people? And he so and so, and he said, he simply said this much: too many players on the field, penalty, all of you get out. So you're not there is even from the clinicians, they don't want anybody else. Yeah, they straight away put a red flag, and that's the thing.
Yeah.
Right.
Okay.
That's fine from a patient perspective, but I think clinicians should still work with the back-end scientists, right?
Correct.
Where you exchange ideas. I know it's changing, it's happening. We just have to catch up. That's all. Correct. I mean, you're right. Like it's the same problem everywhere. Here, they've come to a stage they don't have a choice. They have to do this. And I still face the pushback with people, but I think I have the backing of my like scientific community, right? To move forward. But I see a lot of potential. I think India is the next like the future. I mean, I can see a lot of great things coming out of our country.
India. So let me ask this question. I think we have taken a lot of it. Let this be the last question. Of course, this can be a complete session by itself, but briefly, because you have been one of the first persons who what nowadays we call as women in STEM, in science and technology, and we've been trying hard to get people there to talk more, women scientists to talk more. So we have had mathematicians and others, basically men. So from your perspective, how do you see women's role in science, both in the US and in India? And how has been your own experience? As I said, this should actually have been a complete session, but briefly, if you can.
I mean, to answer your question in a very, very like short, simple manner, I would say, yes, we have made a lot of progress, both in India and in the US.
Yes, okay.
A lot more women are getting into STEM program science, but where both countries, and this is common to both countries, where we have a problem is there are a lot of women initially starting off, and then slowly they just like
paper off.
fizzles away. And that's true in both countries.
Correct.
So I would say that till assistant professor, you'll see a lot of people. Associate, maybe not. Full professor, maybe not that many. Even in big institutions, like any big institution in America and India, it's the same thing. And the pressure that a lot of people talk about, right? The pressure to balance family, still, no matter how progressive a household is, the onus to care for the family falls on the woman, irrespective of country, irrespective of community background, irrespective of that. So I think that is still, women always constantly are making choices. No, I'm going to focus more on this to give up science, because science is brutal. I mean, you have to spend 15 hours a day in the lab to get data, right? So women start making choices slightly as they move up. Again, we have made progress. There is still a lot more that
to be done.
To be done. But I think both countries are same in that sense that we start to see, like the tapering as we go higher up.
Correct.
But we have definitely come a long way. I think we have made tremendous progress.
Because what you say is also something which people always think of. For example, by the time you get to 12th standard results, right, the number of men and women who are in a particular, the top 100 is almost equal or sometimes women dominant. But by the time it comes out of engineering, in a class you have only 20% women, maybe one or two engineerings have slightly more. And by the time you come out to jobs and the managerial position, the vice president position, then it tapers off.
Exactly.
As you said, yeah, there are so many, of course, social issues, and also whether organizations themselves enable this is another question. As I said, it's a huge complex, as complex as our brain now.
No, Suresh, what she says is also true across civilizations.
Yes.
Across countries, women do not get to choose a career over a family, children. So men can have both, women cannot. So it becomes a problem, whether you are from Africa or Asia or America or Europe. So it remains the same. This is a major problem.
Exactly, because US may have made little bit more progress in terms of science and technology, but when it comes to this, we are all the same. Like there's no difference across all countries.
And also that whole concept of the family in US is more explicitly it's told, right? In India, it's more implicit. In every movie that you see of Disney, the final thing is stick with your family, right? Every movie of Disney, that's one message. It's just
right.
across.
And the crazy thing is, in US, in corporate world, if you are a woman and you have a family, I mean, they will just completely, like, of course, they can't obviously come tell you, okay, you have a family, I'm not going to give you a promotion. But it happens all the time. It's one of those things. And particularly in the context of brain diseases, I do want to add that, let's say you have a child with any neurodevelopmental disorder. The stress, the burden that the family has to go through, the father and mother have to go through is enormous. So somebody has to quit a job because you can't have two working parents and a child with seizures all day long, right? And I see this with when I see patients. But again, the onus by default falls on the woman. So that's a separate issue. But the bottom line is, like you both were saying, women always end up choosing. So higher up, it becomes a lot more harder. But it's slowly we'll get there.
Evolving. No, actually, that is true, because in my own where I used to work in a company, there was a woman reporting to me. She was an excellent performer, one of the best performers and all. And unfortunately, her daughter had some autism type of thing. So she had to take a choice at that point in time to quit. No, I mean, there's huge potential for her to grow, but at that point in time, as you said, because of the tension, she had to quit and take care of the child. Now, okay, so I think it's almost hour and a half of your time we have taken.
More than that.
And
thank you. A lot of your time.
So let me let me quickly conclude this. We had an excellent session today with Dr. Surabi, where we spoke about neurogenetics. This is a field which is already started evolving, but has a lot of potential for human beings, because as Dr. Surabi said, and as Rajmohan initially said, brain is something which you have not understood fully. And this is a field which tries to understand that through genes. And then we spoke about what's gene sequencing, what's gene editing, how do we get into trying and solving problems using the genetic sequence, and so on. And Dr. Surabi also told us what they are working on about the early disorders in humans, like when children get autism and others, and how they are trying to attack that, and how even today the treatment is more about symptoms than about the exact disease, because we're still not yet in a position to understand what the disease is, because we see multiple symptoms and we try treating those symptoms. Progress is on, but the progress is slow. And that's one very, very important thing we learned from Dr. Surabi, and I think it's something which a lot of us have to understand is, if you are really in science, you have to, as Feynman once said, never be afraid to admit, never be afraid to admit that you do not know. Saying I do not know is much better than saying that, something false. And, I would personally say, because I don't know Dr. Surabi personally much, but from the interaction, I would say she's the real scientist. So one reason is that she had more than once said, this is our limit of knowledge of this particular field. So we're very, very happy. This is an excellent session. And I thank Dr. Surabi on my behalf and Rajmohan's behalf for sparing this sort of time. And we know that she's an extremely busy person. And all of you would have yourself realized the sort of work that goes into what she's doing. So my personal thanks to you, Dr. Surabi. It's great having you on our show, and we feel privileged that you have joined us.
Thank you. Pleasure is all mine. Thank you so much for this lively interaction. Thank you.
Thank you.
I hope we can do more episodes on related issues. Like one thing that's fascinating me is myth among the neurosurgeons, and myth is not something which is happening because we are all human beings. Like Gazaniga is not dead. Still, there are neurosurgeons who still believe that, and all kinds of motivation talks and all they give across India. Actually, I don't know about other places. There are many aspects to it, social aspects to it.
Yeah.
So we'll do more episodes, hopefully, very soon.
Absolutely. Absolutely. I would love to. Yes, let's do that.
Thank you.
Thank
you very much. Thank you. Bye-bye.